Hello everyone, I'm Dr. Sri Banerjee, core faculty for the College of Health Sciences and Public Policy at Baldwin University, with a joint faculty appointment at Rouse University School of Medicine. In this video, I'll be going over several important research topics that are included part of cancer. I've been saying this in the last couple of days quite a few times, and there's nothing closer to the truth than the idea that in the last 30 years there have not been major contributions that have been made in the field of cancer, which is different than the different specific types of cancer. So for instance, there's certain specific markers which allow certain types of cancer more dangerous. What do I mean by that? This month happens to be Breast Cancer Awareness Month, and when we're thinking about the breast, a lot of times the diagnosis process can be long and drawn out, and unfortunately there's room for a lot of false positives. So the question is, do the people that get mastectomies based on the BRCA1 and BRCA2 gene, does that effectively save breast cancer? So there's a double-edged sword in a lot of these, and when you're thinking about some of the denser breasts, a lot of times in those types of mammograms, it's much more difficult to distinguish between what is cancer and what is dense breast tissue. So differentiating between and understanding the life course and how the human body develops through teenage years, adulthood, and future on. So the oncogenes that are especially important within this context is the end of the beginning of cancer genomics. So this is now going beyond cancer genomics and understanding the oncogenic process. So this is called, by the way, Pan-American Cancer Atlas. It's just a cancer atlas. But very important because the analyses that are highlighting the complex impact of genome alterations on the signaling and multi-omic profiles of human certain cancers, and the influences on tumor microenvironment. So it seems like it's kind of a work in progress in understanding and collaborating, or what ways to collaborate. So this has talked about the TCGA Pre-Cancer Atlas Project, and you can read through the different themes, like somatic driver mutations, germline pathogenic variants, and their interactions. There's some that are quite legitimate. There's some that have extremely important types, genome types, which can be interpreted in the way it should be. And then the substrates for Pan-Cancer Atlas is the associations, driver and molecular subtypes, and immune cell and tumor responses. So all of these facets can be uniquely combined and create the impetus for future research, combining data and analytics and other types of code. So there's a lot of different types of BRCA as being one, but there's a lot of different genomes to be considered. So going back to the, and this is spatial, by the way, the study before. So going back to the flagship paper, there's companion papers in Cell, which talks about germline variants and 10,389 adult cancers. Comprehensive characteristics of cancer driver genes and mutations, driver fusions of their implications in the development and treatment of human cancers. Gao et al. Actually analyzed 10,000 TCGA cohort with 33 cancer types, and the detected gene was fused. They provided a landscape of fusion events detected, related fusions to gene expression, focus on kinase fusion structures, examining mutually exclusive mutations and fusion patterns, and highlight fusion from druggability. What's the immune landscape of cancer while presenting immunogenomics analyses of more than 10,000 tumors identifies 6 immune autotypes that encompass multiple cancer types and are hypothesized to define multiple immune responses patterns impacting programs in the global scale. It's an interesting article to me due to the fact that I typically write a lot of in group a lot of public health components. So we performed meaning different authors. We performed an extensive immunogenic analysis of more than 10,000 tumors, which comprises 33 diverse cancers. And utilizing data compiled by TCGA across cancer types, there are 6 immune subtypes which aided in wound healing. So IFN gamma, dominant, inflammatory, lymphocyte depleted, immunologically quiet is the effect. When we're thinking about TGF beta, which this is characterized by differences in macrophage or lymphocyte signature. Th1, Th2 cell ratios, extent of intratumoral heterogeneity, aneuploidy, extent of neoantigen load, overall cell prediction, proliferation, expression of immunomodulatory genes, and last but not least, prognosis. So the most important portion of of numbers and codes is to be able to make correct informed decisions. This one supports open access, but is not in fact open access, so we may have to look for this in a different place. But just going into this a little bit more, the spatial organization and molecular correlation of tumor infiltrating lymphocytes using deep learning on pathology images. Again, garbage in, garbage out. So the more types of detailed images that are part of a lot of the geospatial components can be included now. So there's genomic and functional approaches to understanding cancer aneuploidy. So now, when we're thinking about going back to the original article, about just thinking about the importance of each of these studies. For instance, this is the PICO model that's been provided. There was a worksheet actually that provides more insight on the PICO model. So that, and the idea is here, this is out of Arizona, is to create evidence-based practice medicine, but then align these with levels of evidence, understanding where your study fits, acquire resource types. So with health information resources, there's systems, but then there's summaries, syntheses, synopses, and studies. And there are examples of a lot of these in Google Scholar, but also the American College of Pediatrics, and different components. So these can be explored, foreground versus background, and then this ends with a well-built patient-oriented questionnaire. I really enjoyed looking at the Arizona College for this, Medical College, and it really shows all of the different parts of this. There's also searching in keywords or subjects. This shows me the process of what you used, and they're called Medline subheadings. And then, of course, you can't go anywhere without actually critically posing appraising the evidence. Sometimes this will iteratively go into the formulating the research question, and you may have to work backwards to conduct a literature review which appropriately aligns with the PICO question that you created and came up with. Quite a few papers go into data analytics, and it is extremely important to understand what the results and why the results are important. How large was the treatment effect? Are results clinically significant, not just statistically significant, or are there additional components which are not known? So during this resource help, the answer to answering my PICO clinical question, will that be enough to write about it? It is important. It is an important article in the particular field. Does it directly address at least one part of the PICO question? Will it help me make my argument? Those are the basic questions. So if you answer them and summarize them, then you can provide a little bit more synopsis for PICO. So this is primary versus secondary sources, and how primary sources, the differences between what types of relationships you're investigating. So when you're thinking about secondary sources, you're thinking about the statistical data that is available at the tip of your fingers. But then also understanding that systematic reviews take time, and in fact, any sort of observational data analysis is conducted by students. And these are very legitimate types of studies, types of research studies, which people can be involved in, engaged in, and oftentimes help individuals retain that residency. So there's a lot of different purposes in conducting a systematic review, meta-analysis, and then at the end there is a fun cartoon which you may or may not find interesting. I hope, again, this has provided you with a foundational insight on how to understand evidence-based practice, even if it doesn't necessarily make sense. Go through all of this, pause my recording, and then you'll definitely find out what I'm saying.